Good morning, everyone, and welcome to day 2 of SfNC 2022. I am Lila Krisiku, and I'm delighted to welcome you to the second day of the conference. Before we kick off the second exciting day of creativity research with our featured speaker presentation, just a couple of very brief notes and reminders. First, everyone should make sure to go vote for the new cover of CRJ at the voting booth in Gavertown. So we'll announce the winner at the end of the conference, and so please make sure that you do that. The link is on the bottom of your screen, the green button. So please do that at some point during the day. And secondly, make sure to stay for the closing remarks at the end of the day for the announcements of conference award winners. So be on the lookout for that. We'll send you some more reminders throughout the day. So without further ado, I'm absolutely delighted to introduce this morning our featured speaker for this year's conference, Dr. Matthijs Baas. Dr. Baas is an associate professor at the Department of Work and Organizational Psychology of the University of Amsterdam, and his research has focused on the cognitive, motivational, and affective foundations of creativity. His work has been particularly influential across many areas of creativity research, including the relationship between mood and creativity, the neural correlates of cognitive flexibility and creative outcomes, and the relationship between creativity and vulnerability to psychopathology. He has authored over 48 peer-reviewed publications, dozens of presentations, and has received many accolades for his teaching and research. We are so excited to hear about the research that you'll be sharing with us today. So, Dr. Baas, the floor is yours.
Thank you so much, Lina. Here we go. So hopefully everybody can see my screen now. So first of all, I'm not a neuroscientist. So I'm a creativity researcher with a background in social and organizational psychology. And it's just that I'm really fascinated with creativity. And one of the reasons is that you can study it from so many disciplines within psychology, including clinical psychology, but also the neuroscience of creativity. You can study it from that perspective as well. And what I would like to do today is basically show how findings and paradigms and theories in these different disciplines can inform us to really get a better understanding of what creativity is. And I will have a special focus on the neuroscience of creativity here. So yesterday, Todd Lubert already mentioned the definition of creativity, and here I present a slightly different one, but it's basically boiling down to the same thing: the generation of ideas, insights, or problem solutions that are new and useful. And Todd was indeed right. You can study this from multiple perspectives. So you have the 7 C's that we learned about yesterday, but you also have the 4 P's. In this case here, I have the person, process, and press perspective. And I think one of the things that are important to realize is that especially the person perspective and the process perspective can be placed in what Kronbach is calling the differential versus experimental psychology. So differential psychology is really about the nature, the cause, and the consequences of psychological differences between individuals. So basically, in terms of the 7 C's, this is about the creator, understanding why some people are more creative than others. So what are these characteristics then differentiating these people? We also have experimental psychology, and that is more focused on the process, the creating in terms of the 7 C's, right? So what happens in the brain during creative problem solving, or what cognitive processes are involved. Now, what you see is if you take the differential perspective, this is about individual differences, what you can then do within neuroscience of creativity is have structural MRI or compare patients with brain damage and then see if somehow people that score high on creativity differ from people that score low in all sorts of cognitive processes or brain processes that you can see. Now, the process perspective is more about measuring process. With fMRI, EEG, PET, what you can also do is manipulating processes with pharmacology, for instance, or TMS. We saw some excellent examples yesterday in various talks. Natasha Mason studying psilocybin, for instance. So these are the differences that I identify. So the person versus process perspective. Now, first, the differential psychology, that is what I turn to first. So this is really about the neurocorrelates of creative ability. And actually, I think that about 10 years ago, Arne Dietrich, in this famous article where he summarized what we then knew about the neuroscience of creativity, had a sort of a gloomy outlook on the field. So there was not really a specific brain area involved in creativity. Also, there were lots of inconsistent findings, difficult to interpret. And I would like to share a few insights that I have on why this will be the case. Now, first thing is that I think there are a lot of methodological challenges that you face when you study creativity. We have these famous tasks that we repeatedly use, the BRIC task, the AUT. What we also use is the RAT, the Remote Associates test by Matthijs. And one of the problems here is that the retest reliability is really below par. So a colleague of mine, Claire Stevenson, what she did is a meta-analysis of the retest reliability of the AUT. So basically the performance on one occasion, whether it predicts the performance on the same sort of test or similar tests on another occasion. And what she found basically is that the correlation between performance on these two occasions on the AUT was a correlation of 0.58. So that means that about 2/3 of the variance is left unexplained. So that also means that it will be very difficult to find reliable differences between people because the very measurement is not so reliable in the first place. Especially when you couple it with typically low statistical power, what you see in many of these studies, because it is very, very difficult to really study these processes with brain procedures. So I think these two are one of the methodological challenges that we face. Now, another is more conceptual in nature, I think. So what is creative ability? So we define it in an article that we wrote as someone's potential to generate real-world creative outcomes. So this is really what you would like to predict. Yeah? Van Gogh coming up with the paintings. And actually, we have a couple of excellent measures for this. So we have the Creative Achievements Questionnaire, James Kaufman, but also Matthijs Benedek came up with all sorts of self-report measures to really capture this criterion that we wish to predict, real-world creative achievements. What we can also do is have people come up with a real creative product, whether it's a poem, whether it's something else, and then these features are typically rated by judges with domain-relevant expertise using the consensual assessment technique by Amabile. What you can also look is really agreed-upon creative feats like patents, awards, prototypes, citations, and I think Dean Simonton offers a couple of really excellent examples of what to look for. But these are the things that you would like to predict in terms of creative ability, right? So the thing is, if you look at the processes that predict these creative outcomes, is that they are a lot. So this is not even an exhaustive list, but you have divergent thinking, convergent thinking, flexibility, persistence, inhibition, set switching, memory retrieval, problem construction. Even if you expand the idea journey a bit, there's an excellent article by Perry Smith and Minucci about this in the Academy of Management Journal. It also says that it doesn't stop with idea generation. Then you need to elaborate on the idea, select the idea, and also champion the idea to others. And that ultimately determines people's success and being creative, really landing a creative achievement. Now, because there are so many processes that predict creative outcomes, what you also see is that there are a lot of individual differences that predict these creative achievements. And this is also not an exhaustive list. So openness to experience, diversifying experience, curiosity, mindfulness skills, ADHD. I mean, the list is so long: positive affectivity, working memory capacity, intelligence. So when you notice this, so there's such a variety in individual differences that are related to creativity. It's not a surprise, I think, that in terms of the findings that Arne Dietrich found in the Psych Bullet article, is that it's very difficult to pinpoint individual differences in the brain related to creativity. So what to do? And I think people in this society already are doing this. So to make creativity tractable in the brain, it must be further subdivided into specific types, different types that can be associated with specific neurocognitive processes. And I think the Ontology project is an excellent example of that. really being more specific, both in the articles that we write, but also in the measures that we use in our research, being more specific, because that ultimately will allow us to really track these different processes in the brain, I think. What we also need is larger samples. That is sometimes very difficult. But if we have these various studies that we have on a particular topic, then ultimately research synthesis will become very, very important, either with a meta-analysis or a systematic review. Actually, there are some famous meta-analysis experts, Hunter and Schmidt, and what they basically say is that each study should be considered an individual data point in a future meta-analysis. And, well, it's a bit of a blunt assessment by these guys, but I do think it is a valuable insight, because I did meta-analysis myself as well. And what you typically see is that there's a large variance in effect sizes regarding a particular topic. In my case, for instance, the mood-creativity relationship. So because of the scattered findings, you need synthesis to make more sense of it and gain a better understanding of what creativity is. Now, what we can also do, I think, is learn from the neuroscience of individual differences that associate with creativity reliably. And to give a bit of an idea of what I mean, I want to focus here on ADHD, something that I researched together with some of my colleagues. So when you look at ADHD, creativity, and neuroscience, what you can do is compare people that score high on ADHD and score low, or people, what you can also do is measure the symptoms that they have. So what is ADHD? It is a neurodevelopmental psychiatric disorder that is characterized by age-inappropriate levels of inattention and or hyperactivity or impulsivity, for instance. And you can just measure these symptoms also in the normal population, right? So what we did in a couple of studies, either measure these symptoms, or what we did is compare people that were clinically diagnosed with ADHD or not. And then we had several of these creativity indicators. And what we typically find there is that people with higher symptoms of ADHD, or people that are characterized by the clinical diagnosis, score higher on creative achievements. When you compare divergent thinking to convergent thinking, we see that ADHD is typically positively related to divergent thinking, but not convergent thinking. Sometimes, but it depends a bit on the measure that we take. Sometimes we see no effect, sometimes we see a negative effect, right? Now, what you can then do is look at what are the genes or what are the brain regions that really characterize people with ADHD. What we then see is that actually genes associated with dopamine or serotonergic or GABA functionality are both involved in creativity. In many studies we find that, but also in ADHD. The same goes for overlap between brain regions such as the PFC and the striatum and networks like the default mode and executive network, what we also find in ADHD and creativity processes. So I think this is very interesting because now we have a body of knowledge both from the individual differences perspective, in this case in terms of ADHD, but also in terms of creativity. And what we now need to do is merge these two in more direct links. For instance, where we compare people with ADHD that are low or high in these symptoms, but simultaneously having them in the scanner, thinking of doing divergent thinking tasks or convergent thinking tasks. And I think this will definitely prove to be very valuable in a better understanding of creativity and the links that we see with these individual differences. So that's one thing. The other thing you can do, and now we move on to the process perspective, the experimental psychology in terms of Kronbach, and this is more about neurocorrelates of the creative process. So here what you can do is measure these processes in terms of EEG and PET or manipulating them. I will turn to that next. Now, here we typically see a clash, right, with the neuroimaging methods and the creativity tasks that we do. So in the neuroimaging methods, what you typically need is many trials. The timing of these processes should be strongly controlled, and we need highly similar tasks for contrast to really make a clean comparison. But that's often very difficult to do with creativity tasks, where typically people have a small number of trials, especially when you take many minutes for coming up with uses for a brick, for instance. The timing of what happens in the brain when you get this creative insight is very unclear. And often the similar tasks for contrast are being used. So this clash is really difficult and something that many of you also experience. So here are a couple of examples where people were asked to come up with, for instance, original uses for a particular object, and they were compared to an instance where people have to come up with ordinary uses or characteristics of the same object. And then you can compare what happens in the brain, right? Here's another example: make a short original sentence, and in the other condition, people needed to do an anagram. Or, for instance, for the Torrance test, people did a divergent thinking task to consequences, and in the other condition, they did a math problem, right? Now, the difficulty here is that there's surely a difference in the creative process that you then trigger, but also in attention or enjoyment, difficulty or mood, or all sorts of things that co-vary and that are very difficult to control and become a confound, potentially. So what we need here, I think, is more specific psychometric instruments, along with a standardized set of control tasks. And this is also what you neuroscientists are very, very good at, because this is, you need to come up with these tasks, right, in order to do your research. So yesterday I saw an interesting talk by Roger Beatty where he said, well, here I have a specific set of words, and people either had to come up with a hypothesis, a generative task, or come up with a synonym, right? And these kinds of things are really cool. And I also think we can move on to crosstalk with other disciplines in psychology where we can use that knowledge. So for instance, in social psychology, what we typically do is treat creativity as the outcome measure. So we're interested in all sorts of variables, independent variables that predict people's creativity, whether it's mood, whether it is something else, whether they are taking more the situational perspective on creativity. What we typically in social psychology and organizational psychology do not do is treat creative thinking as an independent variable. Whether it makes perfect sense to look at this, right? Because creativity, a particular mode, when you think divergently, it can very well impact, for instance, people's perspective taking or stereotyping behavior. And this is really uncharted territory. And I think the expertise that has been developed in neuroscience can definitely be used to inspire research in social psychology, where creative thinking modes can be treated as the independent variable. Really cool. Something to look forward to. This is another example from my own research. So what we did here is indeed focus on more specific types of creative thinking modes. And in this case, we focused on rule convergent versus divergent thinking with an EEG study. Now, divergent thinking, it's typically defined as the generation of multiple alternative solutions in response to an open-ended problem. And what's important here, it relies on a lack of inhibition between alternative thoughts, quick abandoning of rules and examples, and approaching a problem from different perspectives and angles. Now, you can typically contrast this with convergent thinking. Here, there are really, really many definitions, processes, and tasks involved, but it's more perhaps considered to be a syndrome involving many things. So achieving accuracy and correctness, sticking to rules, and sticking to a narrow range of relevant information. Now, in this specific experiment, what we did is we focused on these rules. So the quick abandoning of rules and examples, or sticking to them. And in this case, spontaneous rule convergent versus rule divergent thinking. And the important thing to realize here is that both can ultimately lead to creative outcomes, I think. So rule divergent thinking, everybody has a clear conception of how that works, but with rule convergent thinking, they can also lead to creativity, but this happens more in incremental steps. So the first ideas that people typically come up with are pretty common and mundane, but when you get further within a specific category often, then these ideas tend to get more original. You get past the more mundane ideas. So this is the task that we developed here. We had a lot of fun developing them. The alternative names task. So people were asked to think of new names, for instance, for pasta. This was the category that we then offered. And then what we did is basically we primed people with a certain rule. We cued a certain rule. This was implicit. In this case, Maloveni, Paragoni, and Fellini. And well, this cues the participant that a certain rule, namely a name that you think of, may end with an I, right? So people were asked to come up with new names for various categories, painkillers, bacteria. And then what we did is in the task, people were provided with the category and the examples. Then people were asked to think of a new name. Once they came up with a new name, they pressed the space bar, and then this allowed us to really time lock the moment that people came up with their idea and separate that from the moment where they had to enter their idea, right? And once they entered their idea, they clicked on the space bar again, and had a new opportunity come up with a new name. Now, the cool thing here is what you can do is you can look at the idea that people generated and whether that followed the cue that we primed. So a new pasta name, Snucci, that ended with an I, or that actually diverged from the rule. Grappa, for instance, I mean, it can be anything, right? Now, what we did this EEG experiment, and what we find here, and this goes against many of the findings that we saw before, is that we actually find decreased delta activity for divergent as compared to convergent thinking. So apparently when people thought divergently compared to convergently, we saw a decrease in their delta activity. Now, this is something that I think, yeah, I saw that also in other talks. You have many different frequency bands that are often involved. Many times this is alpha, in our case it's delta. I think we definitely need more research to better understand whether this is a reliable and stable phenomena. For me, it's more that the paradigm that we use can be very interesting because it really allows us to time lock spontaneous, in this case, rule divergent and convergent thinking. And yeah, I would like to see more research with this task and see whether things are replicated. Now, this is just another example here. Besides generation, people can also look at evaluation. Creativity both involves an idea being original and useful. And this is a task that Claire Stevenson is also presenting today at the poster number 239. And we have all sorts of stimulus words here. Using a book as a plate, a boy, or to read, to throw. And from many, many ratings by expert coders, we know that we can classify these ideas in terms of how original they are or useful they are. And what we can then do is present the people with a particular use of a book in this case, and we can ask, okay, do you think this use for a book is creative? Plate. And then the participant is asked, okay, no, not creative, or yes, creative. And we can easily see how we can change these instructions and then relate that, for instance, to all sorts of neuroscientific activations in the brain. And well, actually, there's also already work on that, on how evaluation is correlated with brain activity. Klein-Mintz, Odette has done that. We also, I think, see a talk later today by Heath Matheson. I'm also looking forward to that. But my point here is also move away from idea generation, because I do think that later stages are very interesting as well. Now, this is the final thing you can look at, manipulating process with pharmacology. We already saw some interesting talks also with LSD, for instance, how that impacted creativity. What I did in my research is I focused on oxytocin. Why? Well, because that was available at that time to us to do that research, but also there were some findings that prompted us to look at this. So oxytocin, that hormone that is associated with bonding behavior, is linked to social approach and novelty seeking. And what we do know is that these characteristics are also related to flexibility of thinking and creativity. So what we did here, we had a treatment condition where people either had a nasal spray of oxytocin or a placebo, and then later on, when that kicked in, people did a creativity task. For instance, they generated pasta names here, or in another experiment, they came up with this alternative uses task, for instance, coming up with uses for a brick. Now, what we found here is that oxytocin was indeed associated with more divergent thinking and more flexibility, or more original idea generation. So it was really interesting. And it's also interesting because oxytocin is associated with dopamine and other neuromodulators. And this is how our thinking became a bit more complex and to the point, I would say. And this is a model of the role of dopamine in creative thinking that I generated with a PhD student of mine, Nathalie Boot, and together with Karsten de Dreux, Roshan Kools, and Simon van Gaal. And basically, we distinguish there between different pathways that are involved in dopaminergic processing. So striatal dopamine, which would be related to more flexible kind of thinking, divergent thinking, adopting broad attentional scopes, switching between perspectives. Whereas prefrontal dopamine is more related to persistence, so systematic analysis, convergent thinking, generating ideas within a narrow topic, but these ideas, they get to be more creative over time. And the other insight that we had is perhaps what we see is curvilinear effects. So too much dopamine, striatal dopamine, will lead to distractibility, whereas too much dopamine in prefrontal areas will basically lead to perseveration, rigid thinking. What we need is a balance here between these different regions and the role of dopamine. So we tried to study this. In this case, we looked at methylphenidate, which is typically used for people with ADHD. So we had 48 people, and we had a double-blind placebo-controlled within subject design where people took this medication. And then later on, when it kicked in, they did convergent thinking tasks, the RAT, or divergent thinking tasks. Now, we thought, okay, let's see whether ideas, they actually work. But I've never seen this before, but there were actually flat findings. It reminds me a bit of what Natasha Mason was experiencing yesterday in her talk on the effects of psilocybin. Also, no effect. So it may be that effects were later on emerging, a week later, for instance, but this is not what we tested. What we also considered is perhaps to test this idea that the effects may be curvilinear, is that there may be individual differences in dopaminergic synthesis capacity. And in this case, we know that working memory capacity is associated with the synthesis capacity, but actually also that did not influence the findings. So we're left with lots of questions, and I do think we need more research to get a better idea of what was happening here. And actually, currently what we're doing is actually run a new study with 100 people, could be also a power problem, and also look at pharmacological interventions that focus more on the striatal dopamine, because methylphenidate is more focused on frontal. So this is the final slide. So basically, I think creativity and the brain, there are lots and lots of interesting things happening. Also, I saw that at the conference, at the meeting today and yesterday. But what I do think is what we need is also large sample size studies if we can, and otherwise they should be treated as individual studies that we need to synthesize later, either with systematic reviews or meta-analysis. I think what we're already doing is further subdividing this creativity task into different types along with a standard set of control tasks. And I do think that also can be used for interesting other studies in social psychology, for instance, where we treat creative thinking modes as the independent variable having an impact on all sorts of social psychological phenomena. I also like these more pharmacological intervention studies. We also saw a couple of examples already. And also to consider other stages of the innovation process, like evaluation, selection, perhaps even championing how people react to ideas being presented by others. I think that would be really cool to ultimately get a better understanding of what creative thinking, what it is, and why people differ on this very fascinating topic. And with that, I would like to close with a lot of my collaborators that I dearly work with on these various projects that I just presented. But yeah, thanks for now.
Wonderful. Thank you so much to Matthijs for a very inspiring and thought-provoking talk. We have, I see at least a couple of questions, so let's start with those. So first question, can we infer that the different ways that divergent thinking and convergent thinking deal with rules, either abandon or stick to rules, would possibly explain inconsistent effects of social factors on these two kinds of creative thinking? An interesting question.
So this has to do with the fact that, taking the press perspective, that you have different social factors. Could be. I do think this is one explanation of why that happens, sticking to rules or not, and certain cues that prime people, making them conform more or not. I mean, there's definitely all sorts of variables that you can think of, like social anxiety, that may prompt people. Right? I do think that other explanation can also definitely still exist here, but definitely it's a good idea. Yeah, and then you can focus on this specific phenomena, definitely to see it even as a mediating process, where you establish this rule divergence versus not, or rule sticking or not, to creative performance on a task that follows. Yeah.
Absolutely.
Yeah.
That sounds possible. Yes, of course. Next question, Gregor is asking, you outlined earlier how the diverse elements of creativity and the large number of influencing factors make neurological research difficult, but the field still relies heavily on divergent thinking tasks. How well do you think these findings generalize to other forms of creative expression? And do you think methods need to expand beyond divergent thinking?
I do. So do they generalize? Yeah, it depends a bit on whether there's a common core in what happens during divergent thinking. And as we already saw, divergent thinking in itself is also like a container construct, which involves a lot of things, like changing perspective, easily switching between a particular approach. And you can measure that with different things. Also the switching within an idea generation period, for instance. So even with divergent thinking, it involves many different processes as well. And I think further isolating that is very important to gain a better understanding of what these cognitive processes actually are. Yeah, that makes perfect sense. And I do think that we already do this in our research. Moving beyond divergent thinking, there are many examples in the various talks and posters that we have today where we can learn that. Yeah.
Absolutely. Maybe I'll ask one more question, if you could just elaborate on whether folks should prioritize one of these different areas that you highlighted as being important. We know that there are many different pieces to the complex puzzle, but I'm sure a lot of the members of the audience, and especially the students, are thinking, well, if I'm designing my study, where should I begin? Is there something that you think is, sorely missing, we haven't done as much of, aside from putting the pieces together? Just a speculation.
Where to start? I mean, I do think start with what you're interested in yourself, right? What you're fascinated with. I think there's so many questions that still need answering, or more research to get at a more robust answer. So I think that is important. But what I do know is obviously we tend to stick with divergent thinking perhaps a bit too much. So moving beyond that and also looking at other stages of the creative problem-solving process could be one easy thing to look at, also to some extent, because I think these thoughts may be more suitable to change, to push them in a format where neuroscientific research is easier to do. Yeah.
Wonderful. And I tend to agree. Yes. So thank you to Matthijs for a fantastic talk. I'm sure we'll discuss and think about all the very important points that you've made throughout the day and, of course, beyond. Thank you again so much for joining us this morning. And we'll see everyone in the other room for the sleep session. We'll put the link in the chat.
Yeah.
Thank you again.
Yeah, thank you.